This lecture is focused on fermentation technology, which is a process for the production of microbes. Cellular respiration is the process where organic compounds are converted by enzymes into other products, and this process generates energy in the form of ATP. When the conversion happens in the absence of oxygen, it is called fermentation. The process of fermentation was discovered by Pasteur, who described it as life without air. Anaerobic fermentation pathways are out of the scope of this lecture, but there are many articles and books written on this subject. In biomanufacturing, the term is used differently, so when someone describes a fermentation process, that process can be run with or without oxygen, but as we will see, it is more efficient to run a fermentation with oxygen. For the rest of this lecture, I will use the term fermentation to describe the process, but understand the distinction between respiration and fermentation. In general, fermentation is used to convert low-value products into high-value products. There are lots of products that cells produce that have economic value. These products include cells such as yeast used in the baking process, enzymes and other proteins that are sold commercially, primary metabolites, with one of the best known being ethanol, secondary metabolites such as antibiotics and other chemicals produced by a process called biotransformation, such as steroids. Primary and secondary metabolites differ typically in the timing of their appearance relative to cell growth. Primary metabolites are usually produced as part of metabolism to generate energy and are tied to the growth of cells, while secondary metabolites are not and usually appear later in the growth process. There are also different kinds of fermentation. The simplest form is called batch fermentation, which is essentially putting all of the ingredients to grow cells into one batch. The process is usually less efficient, but most commonly used in research labs. Fed batch is most commonly used in biomanufacturing and works by feeding the cells a nutrient over time to generate large amounts of biomass. The cells need to be fed because adding all of the substrate at once can be detrimental to cells. Perfusion is another type of fermentation and works by having a membrane that separates cells from product, then fresh media is added back. Finally, chemostats are continuous fermentations that work due to growth on a limiting substrate and removal of excess cells to maintain a constant cell density. There are also different types of media that can be used in fermentation. Minimal media, as its name suggests, provides the bare essentials for cell growth and is used for cell labeling of radioisotopes or as a base for fed batch fermentation. Defined media is where the media has a specific chemical composition. Complex media is commonly used in research labs and consists of components such as yeast extract or hydrolyzed protein. Selective media contains a component allowing for growth of one set of microbes and no growth for others. The most common example is the use of antibiotics to select for bacteria the uptake of plasmid containing the corresponding antibiotic resistance gene. Microorganisms grown on differential media display different phenotypes. For example, on McConkey agar, shown in the figure here, contains a dye that is a pH indicator, so in gram-negative bacteria grown, they ferment lactose, causing a change in pH, which results in a change in color to pink in the agar. Equipment that performs the fermentation, which is usually production of microorganisms such as bacteria or fungi, is called a fermenter. There are three types of fermenters, with the most common being stirred tank. We will get into the details on this in later lectures, but it consists of a stainless steel or glass tank as shown on the left in the figure, which is stirred from the inside in order to mix components and deliver oxygen to cells. Bioreactors, like the whey bioreactor on the right, have a similar function as fermenters, but are called as such when they are used to produce mammalian cells. Whey bioreactors, as the name suggests, rock back and forth, creating a wave-like motion which performs the mixing and oxygenation function. These are typically used to produce cells for biopharmaceutical products or as seed tanks for larger bioreactors. Airlift bioreactors use gas bubbles to mix and oxygenate the vessel, which results in less shear stress than fermenters and are used for growing shear-sensitive cells such as plant cells. Where do the strains we grow in fermenters come from? Some strains are naturally occurring, although only about 1% of environmental isolates are culturable. Strains that have been identified become part of culture collections such as the American Type Culture Collection, or ATCC. There are hundreds of E. coli strains, some of which are found in the E. coli Genetic Stock Center, or CGSC, at Yale. Fungal strains can be found in the Fungal Genetic Stock Center, and there are many other sources where researchers can order strains. The other source of strains are novel strains created in the lab using organisms that are genetically alterable, such as E. coli. Plasmids can be transformed into this bacteria, creating strains able to express proteins from other organisms, or whole pathways that result in new chemicals produced by cells. Without selection, E. coli can lose its plasmid, so other methods have been developed to create what are known as stable cell lines, both for bacteria and for mammalian and fungal cells, where the genes of interest are integrated into the chromosome of the cell. As mentioned in the last slide, plasmids can be used to introduce novel DNA into cells. There are several elements that are necessary for protein expression. These elements include a ribosome binding site, promoter, and other elements such as the origin of replication to propagate the plasmid, and an antibiotic resistance gene for selection. Once the strain has been chosen, then production can begin. There are 4 phases of cell growth. The first is lag phase, which can vary in length but is essentially a time when the cells are ramping up to start dividing. Cell division occurs in the next phase called exponential phase, which is characterized by a rapid increase in cell numbers as they are growing exponentially. Stationary phase occurs once nutrients are depleted or other factors cause cells to stop dividing. This is followed by death phase, in which cell numbers start to decline as few new cells are produced to replace those that have died. These phases are usually tracked by the total cell number using measurements of optical density of the culture. Cell numbers can be estimated by measuring the light absorbed at wavelengths around 550 to 600 nanometers. As shown in the figure, this works by shining light through a culture filtered to the desired wavelength, which is then recorded. For standard spectrophotometers, the usable limit of detection is around 1 optical density, or OD. After this, cultures should be diluted in order to calculate the final OD. Another type of instrument to estimate cell growth is a cletmeter. These photometric colorimeters use specific light filters to give readings on a unique clet scale. The readings are directly proportional to the cell concentration or optical density of a culture. Because of cell division, microbial growth is exponential, as shown in the displayed graph. To calculate cell numbers at generation n, multiply the initial cell number of n sub 0 by 2 to the power of the total generations. The time between generations is called doubling time, and it is described by the formula at the bottom of the slide. Another way to calculate the specific growth rate, or mu as it is commonly referred to, is to divide the natural log of 2, which is approximately 0.693, and the slope of the exponential curve. In the example shown in this slide, the calculated slope is 0.0358, so the doubling time in this case would be 19.36 minutes. In order for cells to grow, they need nutrients. As shown in the table here, carbon, oxygen, and nitrogen are the most abundant and are the main consideration in media formulations. Other components such as sulfur, magnesium, calcium, and iron play a role as cofactors and components of proteins. These nutritional requirements serve as the electron acceptors that are part of the metabolism of organisms and result in energy production. The formulation of these components have a direct effect on fermentation yields, as we'll see in later lectures. As was mentioned earlier, rich media is commonly used in research labs and in seed culture for larger scale fermentations. This media contains all of the nutrients required for growth and are comprised of proteins and protein fragments, along with other factors. Rich media supports robust growth because cells don't have to manufacture key components for growth, as they can be directly derived from the media. There are several types of rich media, including casein, tryptone, peptones, and yeast extract. These components are formulated into different media recipes that have been developed over time. These recipes include LB, Luria broth, and the richer formulations including super broth, terrific broth, and YPD. Some of these are buffered as acid production can occur because of excess nutrients being introduced in a batch versus being fed as mentioned previously. Originally derived from animal sources, most of the rich media products are plant-based in order to meet regulatory requirements that disallow animal components because of risk of viral or other contaminants. Another development is the use of self or auto-inducing media, which does not require the operator to monitor the optical density of the culture, as induction is initiated after a key nutrient has been used up. Rich media has several advantages over other media, including resulting in higher yields and faster growth rates. The growth is very reliable, and there are lots of data on use of rich media and growth rates for many organisms. Rich media has some disadvantages, mainly due to the fast growth rates achieved. This can result in aberrant protein expression, causing inclusion body formation and other protein degradation. In addition, complex media introduces a heterogeneous mixture of proteins which have to be dealt with later during downstream processing. In addition, other challenges exist in scale-up fermentation from use of rich media. These include loss of ability to control oxygen and higher demand for cooling due to excessive heating of cultures. In addition, there can be batch-to-batch variability in media lots and between vendors, so it is recommended to test batches before scaling up. For this reason, use of defined media may be preferred as growth rates can be limited by addition of a key nutrient to control the growth rate. This is typically achieved through the use of fed batch or continuous strategies. In conclusion, the primary aim of fermentation is maximizing product yield. This may not necessarily be biomass because the two are not always linked.